Hello doctors, welcome back to Pirates of Medicine. So in today's video we are going to continue with the biochemistry base. Before we start, I would like to thank you all for giving so much love to our videos. I've got various suggestions and reviews, and to talk about it a little, we are trying really hard to prepare these bases as fast and as quick as we can. But if there is certain delay or it takes time, please bear with us and continue loving our videos and sharing our videos like you are doing right now. So thank you. Anyway, let's begin without any further delay. Okay, so the 21st question is that a 4-year-old child with signs of proteinic starvation was admitted to the hospital. There were signs of growth inhibition, anemia, edemata, mental deficiency. So they're asking what is the cause of this edemata development. So edema, we all know, edema is swelling of any part of the body. So to understand this concept, let's go to the basics. What albumin is. So albumin, first of all, is a plasma protein. It is the main protein of the human blood plasma. It makes up about 50% of human plasma protein. Now albumin has some interesting functions. First of all, it grasps everything around it and keeps everything to itself. Like it binds water, it binds cations such as calcium, sodium and potassium, it binds fatty acids, hormones, bilirubin, even thyroxine hormones, and even certain drugs like barbiturates as well. So it binds these substances and keeps them from seeping into the extracellular fluid. So another function of albumin is that it maintains the oncotic pressure of the blood. It also has something to do with the coagulation mechanism and also maintains the intravascular pressure as well. So what happens if we have a deficiency of albumins in our body? The condition is known as hypoalbuminemia. So it is a medical sign in which the level of albumin in the blood is low, and this can be due to either it can be due to the decreased production in the liver, or it can be due to increased loss in the GIT or the kidneys, or it can also be due to increased use in the body, and also due to abnormal distribution between body compartments. So all these are the etiologies that can cause hypoalbuminemia in the body. Now, what happens if we have hypoalbuminemia? What happens is that this leads to an abnormal distribution of fluids because the albumins are not there to hold water and other substances close to themselves. This results in edemas, specifically in the lower legs or the lower limbs, ascites in the abdomen, and also effusions around the cavities near the internal organs. So this is a classic manifestation of hypoalbuminemia: edema, ascites, and effusion around the internal organs. So the edema development causes reduced synthesis of albumins, causing hypoalbuminemia. Let's go to the next one. So in 22nd, researchers isolated 5 isoenzymic forms of lactate dehydrogenase from the human blood serum, and they studied their properties. What property indicates that the isoenzymic form were isolated from the same enzyme? Isoenzyme. Isoenzymes are enzymes that differ in the amino acid sequence, but they catalyze the same chemical reaction. The most important thing that you have to get in this concept is isoenzymes are the same enzymes. They are like isomers that we studied during our 12th standard biochemistry or bioorganic chemistry specifically. So these are isomers which differ in specific amino acid sequence, but they catalyze the same reaction. Now because of this difference in amino acid sequence, they have some dissimilarities as well. Like they have different kinetic parameters like different Km, different Vmax value, different half-lives, etc. You know, that thing we studied in enzymes. So and also they have different regulatory properties. But the most important thing, and I'm stressing it again, they catalyze the same reaction always. So all the isoenzymes of lactate dehydrogenase, they catalyze the same reaction in which lactate is converted into pyruvate and or the pyruvate is converted into lactate. So if we talk about the isoenzyme of the lactate dehydrogenase, the lactate dehydrogenase is itself a tetramer formed of different subunits. So isoenzymic forms are different in these subunits. For example, the first isoenzyme of LDH will have H4 subunits, and it is located in the heart and erythrocyte. The second form has one different subunit with the H, that is the M subunit, so it's called H3M, and it is again located in the heart and erythrocytes. LDH3 has 2 M units and 2 H units, and it is located in brain and kidney. LDH4 on the other hand has 3 M units, HM3 and 1 H unit, and it is located in skeletal muscles and liver. LDH5 M4 skeletal muscle and liver. Now the importance of this isoenzymes is that even though they catalyze the same reaction, they have certain diagnostic importance. For example, I give you an example. In case of myocardial infarction, when there is an injury to the heart or the cardiac tissue, the cardiac tissue will be, or the specifically cardiac cell will be disintegrated, and the enzymes in the cell will be released into the blood. So one such enzyme is LDH1 or even LDH2. So if there is an increase in amount of LDH1 in the blood, it can be used as a diagnostic marker for myocardial infarction or any heart disease, because in that case only LDH1 will be increased, not LDH3, 4 or 5. Correct? Similarly, if there is a liver disease, LDH4 or 5 can be increased, giving us that diagnostic marker. So I think that covers it. So on 23rd question, on some diseases it is observed aldosteronism with hypertension and edema due to sodium retention in the body. What organ of the internal secretion is affected on aldosteronism? So to understand aldosteronism, let's talk about aldosterone. Aldosterone is a hormone which is synthesized in the body from corticosterone, which is a steroid derivative from cholesterol. Now this production of aldosterone, it occurs in the zona glomerulosa of the adrenal cortex, and it is regulated by the renin-angiotensin mechanism. So you all know the function of aldosterone. It is sodium retention in the body. So this hormone goes to the kidney and it instructs the nephrons to retain the sodium ions and not excreted in the urine. So aldosteronism is a condition where this excessive secretion of aldosterone, which results in the disturbance of balance of sodium, potassium and water, and effectively leads to high blood pressure. Now there are 3 types of aldosteronism. This primary, secondary and pseudo hyperaldosteronism. Primary is due to a aldosterone secreting tumor or congenital adrenal hyperplasia. These tumors are located in the adrenal cortex itself. In this condition, there is high aldosterone, but the amount of renin is almost normal or low. In secondary hyperaldosteronism, it occurs due to a secondary condition not in the renal cortex, not in the adrenal cortex. So it may be due to a renovascular disease which disturbs the renin-angiotensin mechanism or a renin secreting tumor. So in this case, renin will be high, and as a result, aldosterone will be high. Pseudo hyperaldosteronism is like a concept in which there is a Cushing syndrome or exogenous mineralocorticoids that leads to indirectly increase in aldosterone levels, which are almost normal or slightly low. So it is actually a condition in which aldosterone levels are normal, but it imitates as if there is sodium retention, which is classically seen in Cushing syndrome. So I think that covers this. So 24th question is that an experiment proved that UV radiated cells of patients with xeroderma pigmentosum restored the native DNA structure slower than the cells of healthy individuals as a result of reparation enzyme defection. What enzyme helps in this process. So to understand this, let's take a quick look at the DNA repair mechanisms. So first we have base excision repair. In this specifically, you can understand from the name, a base excision, that a removal of a base. If a single base of the DNA is damaged, then this enzyme and glycosylase can remove this base and then give a new one and repair this damage. Then nucleotide excision. If a nucleotide sequence is damaged, then an enzyme called as excision endonuclease removes this fragment and joins and replaces it. Mismatch repair is a type of DNA damage in which it occurs due to copying errors. So the strand is removed by exonuclease and then it is replaced. Then we have double strand break repair, which is a little complex. In this unwinding, alignment and ligation, these processes occur. So xeroderma pigmentosum is caused by cellular hypersensitivity to ultraviolet radiation as a result of a defect in DNA repair system. Now, what is this defect? First of all, it is inherited in an autosomal recessive manner. It is a hereditary disease. Now, what is the damage or the defect in the DNA repair system? It occurs due to a deficiency or damage to this enzyme called as excision endonuclease, which I told you is responsible for nucleotide excision repair. Now, if this enzyme is mutated or damaged, then the damage to the DNA which occurs due to exposure to the UV light is not done, which leads to the classic symptoms of xeroderma pigmentosum. Freckles, photophobia, skin cancer can occur, tumor of eye, premature skin aging, and certain neurological problems as well. So 25th question is that a patient has suspicion of epidemic typhus. He was admitted to the hospital. There are some arachnids and insects found in his flat. Which of them may be a carrier of the pathogen of epidemic typhus? So epidemic typhus is also called as louse bone or lice bone typhus. It is caused by the microorganism Rickettsia provozeki and is transmitted to the infected feces of the insect or the vector body lice Pediculus humanus. Now, louse-borne typhus is responsible for large epidemics, which we saw in the question, in population with poor sanitary and overcrowded conditions. Look at this poor guy. Anyway, so the symptoms that are seen is that there is high fever and chills, there is generalized myalgia or muscle pain, there is a macular rash like we see in this guy. After 4 to 7 days, the patient may lack consciousness, which I think is seen in this guy, poor soul, and also severe headache. So I think that pretty much answers it, that the vector is lice. So 26th question is that a businessman came to India from South America. On examination, the physician found that the patient was suffering from sleeping sickness. What was the way of invasion? Now, American Trypanosomiasis or Chagas disease is an insect-borne parasitic infection of humans and other animals. So it is quite common in South America, correct? So it is also called as American sleeping sickness. In this, the parasite is Trypanosoma cruzi specifically, and the vector that transfers Trypanosoma cruzi from human to human is Triatoma infestans, or also called as kissing bug, which we can see this guy to the right, pretty scary looking guy. So the clinical manifestations of American Trypanosomiasis is there is first of all an acute stage and then a chronic stage. In acute stage, there are some general symptoms like asymptomatic or mild symptoms like lymphadenopathy, fever, headache, myalgia, abdominal pain. In extreme cases of acute myocarditis and meningoencephalitis can cause death. If this disease occurs for a chronic period, then some cardiac symptoms can develop like acute congestive heart failure, myocarditis, arrhythmias. Some GIT symptoms are common like mesoesophagus, regurgitation, megacolon with constipation, etc. The diagnosis is just examining the blood, fluid, bone marrow, lymph node aspirate or the CSF under a microscope. We can clearly see Trypanosoma cruzi under a microscope, this little guy to the right. The treatment is benzenazole or lampit, which are commonly used for the treatment. So 27th question is that there is observed inhibited fibrillation in the patient with bile duct obstruction, bleeding due to low level of absorption of some vitamin. So what vitamin is this? So because of bile duct obstruction, there will be low secretion of bile into the intestine. Now there are 4 bile dependent fat soluble vitamins, which are vitamin A, D, E and K, the first 4 in our options. So which of these vitamins can cause bleeding if they are deficient? That would be our special vitamin K. So it is the fat soluble vitamin. It is the only fat soluble vitamin that has a coenzyme function. So either it is taken by diet or it can be synthesized by bacteria. It exists in 3 forms. What is the function? It acts as a coenzyme for carboxylation of glutamic acid residues present in precursor of clotting factors. So this is a precursor clotting factor, and you see glutamic coming, glutamic acid coming over here. Now what happens is carboxylase, this enzyme with the help of vitamin K as a coenzyme, it carboxylates this glutamic acid so that this structure has 2 carboxylic group, which has further action which we will talk about later. But this is the main mechanism in which vitamin K acts as a coenzyme for carboxylation of glutamic acid. Now the deficiency of vitamin K obviously will lead to blood clotting time being increased and disturbances in blood clotting, and hemorrhages can occur as result. Now there is also hypervitaminosis of vitamin K, which can cause hemolytic anemia and jaundice due to increased lysis of red blood cells. 28th question is that a 52-year-old patient with bronchial asthma was treated with glucocorticoids. Fever reaction appeared as a result of post-injective abscess. The patient had subfebrile temperature, which didn't correspond to the latitude and severity of inflammatory process. Why did patient have low fever reaction? So the thing to notice is that the patient has an abscess. An abscess is a cavity filled with inflammatory exudate. So if an abscess occurs in a patient, it must mean that there is a high inflammation reaction going on this patient. And one of the five clinical morphological symptoms of inflammation is edema formation, redness, pain, increased temperature and loss of function. So why isn't there an increase of temperature in this case? Why isn't there fever in this case? Because the patient has subfebrile temperature. So we notice that the patient has been treated with glucocorticoids. What do glucocorticoids do? So there are some endogenous pyrogens which we can say these are prostaglandins or cytokines. So what happens is that glucocorticoid, they inhibit the COX or the LOX cycle at its base. There are these membrane phospholipids. They are converted into arachidonic acid by the help of enzyme phospholipase A2, and this way the COX cycle continues towards the left, and if the COX cycle is inhibited, then the LOX cycle continues. They occur whenever they are needed. So if the COX enzyme, the cyclooxygenase enzyme, it converts the arachidonic acid to PGG2, which is a precursor of prostaglandin. Then they are converted into respective prostaglandins, and they continue doing their functions. Similarly, in LOX pathway, lipoxygenase converts arachidonic acid to 5-HPETE, which converts further into leukotrienes. And these prostaglandins are these endogenic pyrogens which go to the hypothalamus and signal it to please increase the temperature. Now, if glucocorticoids inhibit phospholipase A2, there would be no arachidonic acid and no endogenous pyrogen will be formed. So in this case, glucocorticoid has inhibited endogenous pyrogen production. This way, the patient's temperature is subfebrile and does not correspond to the latitude and severity of the inflammatory process. Another part is that we'll learn about it later also, that there are these non-steroidal anti-inflammatory drugs which inhibit Krok enzymes which help us to treat inflammation. So let's go to the next one. A 35-year-old patient or man under the treatment for pulmonary tuberculosis has acute onset of right big toe pain, swelling and low-grade fever. The gouty arthritis was diagnosed and high serum uric acid was found. Which of the anti-TB drugs are known for causing uric acid levels? So pyrazinamide, our friend for today, is the medication used to treat acute form of tuberculosis. The mechanism of action is that it diffuses into the Mycobacterium tuberculosis and which has this enzyme called as pyracinamase, which converts the pyrazinamide to its active form, which is pyrazinoic acid. This pyrazinoic acid inhibits the mycolic acid inhibition through a cascade of mechanisms and then also disrupts the cell membrane integrity. This is the mechanism of action. Now, what are these side effects? Pyrazinamide has classic side effect of fever and an important hyperuricemia which can cause gout. The most dangerous effect is hepatotoxicity. And there are some other symptoms like nausea and vomiting, arthralgia, myalgia, thrombocytopenia, anemia. You need to remember pyrazinamide causes hyperuricemia or gout, or it can also cause hepatotoxicity. So mostly pyrazinamide is given with combination with certain other drugs, anti-TB drugs, rifampicin, ethambutol, isoniazid. So pyrazinamide can cause high uric acid levels. 30th question is that during the metabolic process, active forms of the oxygen including superoxide and anion radical are formed. With what help of enzyme is this anion activated? So what happens is that during some metabolic reaction, cell produce superoxide ions by a reaction called catalyzed by NADPH reductase. Now this superoxide radical along with the hypochlorous ions, they bring about bactericidal action in case of phagocytes, which we are going to talk right here. So this is the role of superoxide ions in phagocytosis. Anyway, this free radical formation which is seen during the electron transport chain and even in phagocytosis, these are capable of damaging almost all the types of biomolecules like protein, lipids, carbohydrates and nucleoacids. What will happen if these biomolecules are damaged? These biomolecules, they make up the whole structure of our body. So damage to these biomolecules will cause extensive damage to our cells. So imagine this superoxide ion being formed here, and what happens next is that it is converted into hydrogen peroxide by superoxide dismutase, and myeloperoxidase converts it into hypochlorous acid. So these three are the reactive oxygen species which can cause damage to the body. Now to save us from that damage, there are certain antioxidants present in the body which are synthesized by the body and which we take as nutrients. So first of all, we have enzymes that act as antioxidants. We have superoxide dismutase, which converts the superoxide ion back to hydrogen peroxide. Then we have the catalase, which converts the hydrogen peroxide form into water and oxygen. Then we have glutathione peroxidase and glutathione reductase, which are a part of the glutathione complex, which converts hydrogen peroxide back to water and takes NADPH for this mechanism. So these are the enzyme antioxidants. We have certain non-enzymatic antioxidants as well. The carotenoids, beta carotene, alpha tocopherol, ascorbic acid, vitamin C, selenium, metal, iron. And also there are some metabolic antioxidants like glutathione, ceruloplasmin, albumin, bilirubin, transferrin, ferritin, and uric acid. So the question was, which help this anion is deactivated? It is this is superoxide ion, which is being deactivated by superoxide dismutase. Okay, so I think that's all for today. If you like this video, please click that like button, and if you hadn't already, please subscribe to the channel. And if you found the last couple of videos, please share these videos with your friends and the other Krok aspirants. We'll try to post more videos as quick as we can. So until then, it's been good. See you in the next one.